Yeah, no worries. Appreciate it. Thank you, Goran. Good morning, good afternoon, good evening, everyone, wherever you are in the world. Welcome to the Virtual Physiological Human Institute Keynote Webinar Series. My name is Yogesh Kumaran. I'm an MD PhD candidate at the University of Toledo in the United States and a member of the VPHi Young Scientist Committee. A couple of housekeeping things before we get started. The session will be recorded and on the VPHi website likely later this week. So I'll do a brief introduction, 45-minute keynote, and then leave about 10 to 15 minutes of Q&A towards the end. When we do get to the Q&A session, please raise your hand and we can unmute you, or you can feel free to type your questions in the questions chat at any time, and we can curate them for discussion. With that, it is my pleasure to introduce Professor Jose Manuel Garcia Aznar, full professor of mechanical engineering at the University of Zaragoza. His career has spanned over 30 years, ranging from works from multiscale tumor growth modeling to skeletal tissue regeneration and hybrid in silico microfluidic experimentation. Today, we have the pleasure of hearing his talk, Challenges in the Multiscale Modeling of Tumor Growth, from patient-specific models to tumor organoids. Please join me in welcoming Professor Garcia Aznar. Professor, the floor is yours.
Okay, thank you very much for your kind presentation. Thank you very much for giving me the opportunity to present the work that we are developing here in Zaragoza. So I have focused my presentation in the challenges that we have to face to modeling tumor growth. So aligned with this topic, cancer, most of us know that is a group of diseases that are characterized by a high complexity. So this complexity have many implications, because to understand how these processes require different kind of technology. So our idea is to combine different kind of modeling approaches. We have you can see here in my slide on the right hand, different kind of experimental model where we have different data. But our idea is to combine this data with computational models. The advantage of in silico models is that we can obtain a lot of information, but of course we need to validate this information with experimental data. So my presentation is going to focus in the relevance of validation of our models. So in this context, computational model computational modeling can be organized or structured using different techniques or different approaches. So how we have defined these approaches? It's clear that in cancer and in general in biology, the multiscale and hybrid approach are the tendency. But of course, they are even in keeping in mind this approach, we have different possibilities and different strategies that can be used. So the main challenges is we have to select, when we are interested in simulating a problem in biology, we need to define the modeling approach. We have different strategies, continuous, discrete, or hybrid. But also we need to construct the model. We can construct the model using image-based data, but also we need to define if we are going to use individual or distribution of cells. We need to select which is going to be the numerical solver, finite element, finite differences, etc. And of course, we need to verify that our code is valid for the simulation that we would like to do. And finally, what is, in my opinion, more relevant in any problem of biology is validation. Validation is crucial. And in my opinion, validation have to define the kind of model that we have to simulate. If you don't mind, I can indicate more data about validation. Validation is very useful. You observe in the literature, there are many models. Just Box said, all models are wrong, but some are useful. Following the review paper of Markowitz, he indicated that most of published clinical models are never used in clinic because the problem is most of them never have been validated. So only few models have been validated. So in my opinion, the validation is crucial. It's not always necessary to have data only from our lab, but we can use data available in routine practice. This is the most important, to have data that normally clinicians are using in their routine practice. So with this idea in mind, in my opinion, validation is the process that drives the modeling strategy. So the modeling strategy has to be driven by the validation. So validation is addressing how to be define the modeling approach. So with this idea in mind, I would like to show this strategy that we are trying to use, depending on the sources of data that we have. Initially, I'm going to focus in vitro experiments that we are developing in our lab. But also I would like to show other data that obtained from other labs. So I focus my presentation in tumor organoids that normally are cultured in vitro. So normally tumor organoids are 3D self-organized structures that are derived from specific tissue samples. So they try to recreate the tumor in vitro. So we are using tumor organoids cultured in microfluidic chips because they provide an easy way to quantify the size and the volume of these organoids and also their architecture. So actually, we culture, normally what we do is we culture individual cells in the chip and we give them, depending on the stiffness, we give them the possibility to these tumor cells to grow to form tumor spheroids. So you can visualize here an animation where, starting with only individual cells, we can observe how this tumor is growing. So we can obtain data from this tumor, like cell organization, size, etc. So we need data. So from this data, we are going to do some experiments. So in this first case, we have data from other authors. In this case, they culture mammary tumor cells. So you can observe that in this case, these organoids, when they culture these organoids in stiffness, in elastic modulus, in gels of a low value, we can observe that the morphology of the organoid is quite healthy, with a lumen and with cells surrounding this lumen. However, when we increase the stiffness, when these authors increase the stiffness of the elastic of the hydrogel, is a collagen gel, increasing the concentration of the collagen, they observe that there is no one lumen. There are multiple lumens and different cells. So the architecture of the organoid is completely wrong. It's more similar to an adenocarcinoma. So our first idea is to use this data. But additionally, we have developed more experiments to obtain quantitative data of this process. In this case, we use other cell types, but we culture cell organoids and we evaluate not only the number of cells, but also the lumen volume. So the numerical model is focusing modeling this tumor organoid, but with different type of data. Initially, I'm going to focus in modeling cystic organoid, but only with qualitative data. So in this case, this is the modeling that we define. We are going to use an agent-based model using a center-based approach, assuming non-deformable cell. One agent is one cell. So we construct the model with the idea to simulate this process in which initially we have two cells. These cells proliferate and later there is a reversal process, and in this area we are going to have directly the formation of the lumen that we can see here. This lumen is due because these cells are secreting fluid inside the lumen. So to simulate this, we construct a model based on a ion-based approach where we keep in mind different approach. We keep in the interaction between the fluid and the cells. We take into account the cell-cell interaction. We also simulate the interaction of the matrix with the cell. And finally, cell proliferation. All these phenomena have been included in our model. With this idea in mind, we construct a model that keep in mind or take into account the cell cycle. Initially, if the cell is polarized or not. Depending on that, it produces the division. Later, the cell, if it's active, is going to proliferate, and if it's not active, it keeps quiet. Of course, we are going to take into account the fluid secretion and additionally all the mechanical interaction between cell with matrix, and cell with fluid. With this approach, we construct a physical and mechanical model where we take into account the equilibrium equation for the cells and also for the fluid particles, because we simulate the fluid also using a particle-based approach. And the fluid secretion at each lumen is depending on the luminal hydrostatic pressure. So the cells are secreting fluid. Of course, before simulating this process, we verify that our model is valid. It's valid to simulate a fluid. So for that, we conducted a shear test to check, to verify that our approach really simulates an actual fluid. And additionally, we simulate different conditions. It's possible to create only one lumen, as we can see in the first case, or in the case that we have different lumens that can create 2 lumens or even more lumens. So you can see here that our model is able to simulate the polarization, the mid-body formation, the preapical, and you can visualize here in shadow the formation of the lumen. Sorry, now this, okay, now, okay, you can visualize the fluid secretion and the process. So what we did is we simulate different properties of the matrix, which change according to the publication of PASEC. We modify the mechanical properties of the surrounding matrix in the organoid. And we can observe that our model is able to, in the case of using a low density matrix, we obtain an organoid with a single regular lumen that is very physiological. However, when we increase the density of the matrix, we can observe multiple lumens, an aberrant architecture that is very similar what experimentally was observed in the experiment. So we obtain an architecture that is more similar to a carcinoma, a tumor. So this sample of modeling was mainly focused in a qualitative validation. We are not interested exactly to determine how many, but is to predict the tendency of this. However, we would like to go a step forward, obtaining a more quantitative validation. In this case, we focus in pancreatic ductal adenocarcinoma, and these cells were cultured in matrigel. We can observe on the right in the simulation that how in vitro this organoid is formed. But what we observed that in the same, exactly in the same condition, sorry, in the same condition, we select 4 organoids, but we can observe that there is a large variability. So we quantify the number of cells and the lumen volume. And for example, we observe the organoid 4 is much bigger than the other organoids. The organoid 3 is much smaller. And later, organoid 1 and 2 are more intermediate. So in this case, in the same, exactly the same conditions, we obtain different kind of organoids. So we try with the simulation, try to understand what is happening in this case. So keeping the same approach, we try to go to give a step forward. So the cells is clear that are sensing different kind of stimulus. And these sensing are transduced. And finally, the cell give a response. This response can be proliferate, migration, secretion of fluid or not. Of course, this process is very complex. It's very complex because we have different biochemical reaction, probability states. So our idea is to simplify this process and simulate the cell response depending on the stimulus as a neural network. So the idea is to combine, on the one hand, a physics-based model that is using agent-based models, and additionally, keep in mind the experimental data. So this combination of experimental data and the physics-based model, we are going to create a neural network that is feeding our model, numerical model is feeding from the experimental data. So we can understand how the morphology of the organoid is evolving, is temporally evolving. So with this idea in mind, what we are doing is, on the one hand, we have the experimental data. We have quantified, and we know the number of cells and the lumen volume. So with this, we define a simulation metrics, and we are simulating with our model the different conditions. We simulate proliferation, we simulate different processes, okay? But in any case, sorry, we have to define a fitness function. This fitness function is going to define if our numerical simulation is or not a good prediction of the experimental result. So what we do later is to fit and to train this neural network. So in the vertical axis, we present the fitness. The fitness is the solution, quantifies our solution, our numerical solution is good or not in comparison with the experimental result. And in the horizontal axis, we represent the different iteration. So we can observe that in the first iteration, the first generation of the neural network, our organoid is not good because it doesn't have a lumen. So the neural network have to learn how to create lumen. So if we repeat the process, in the generation 100, we can observe that our neural network has learned how to create lumen, but the lumen is not good because it's in the boundary, it's not exactly in the center of the organoid. So we need to improve and to increase the fitness. We need to keep learning of training the neural network. And we can observe that little by little, the lumen is formed in the middle, in the center of the organoid. Finally, in the 400, we can see that the convergence is increasing. And finally, in this condition, more or less are similar. So this system has learned how to create an organoid. This neural network is a representation of how the cells are able to create this lumen. So we quantify the 4 organoids that we have, and we observe that each organoid has followed a different morphogenetic pattern. So how we can evaluate it? We represent in this figure the in blue is the proliferation cells, so when the cells are proliferating. In red, we show how the cells are secreting, And in green, we are showing how the cells are kinescent. For example, we can observe that the organoid that is larger requires much more time proliferating, and however, the secretion occurs later in the time. However, the organoid that is smaller is proliferating very shortly, and the secretion of fluid is very short in time, and later they proliferate again, but later all the cells keep quiescent. However, in the intermediate organoids, the process is, of course, an intermediate case. So initially they proliferate, and later the fluid secretion occurs later than in the smaller, but before than in the larger. So clearly, this is a simple example, but we can observe that we can find an explanation of why we obtain different morphogenetic pattern, different variability in our organoids. Of course, later we have all the simulations for example, in this case is the first organoid, but we can simulate or we can obtain any kind of organoid under these conditions. After this first example that I have focused in the simulation of in vitro experiments of tumor organoid, I'm going to focus in a second example that we have developed, in this case using clinical data. So our objective is the objective of the project ProCanate is to create pattern-specific models to predict the prostate tumor. So the idea is to create a digital twin of the tumor of the pattern to predict how the tumor is going to evolve, is going to progress with the time. So of course, for predict, to evaluate it, we have data from the patient, we have magnetic resonance imaging data, and we are going to have PCA test, PCA data to evaluate how the progression of the tumor is. So in this case, we need to define, of course, a different approach because we have different kind of data and different kind of information. So our modeling approach is based in a continuum approach. We have a continuum tumor cell concentration, and we are going to use basically partial differential equation and ordinary differential equation. For construct the model, we need to obtain the image data. So using the MRI data, we are going to construct the model, trying to create a model that is representative of the tumor of the patient, of the prostate of the patient. For this, we use the different data, the ADC map. From the ADC map, we are able to obtain the cellularity map in the tumor. From the DCA, we need different kind of numerical approach to estimate how is the vascularization map. So we use different numerical models that is based in different technologies, but at the end we are able to obtain a vascularization map. And finally, with the data from MRI, we are able to create a smooth 3D reconstruction and the finite element model. So in the model, we are going to have the cellular distribution, the vascular distribution. So with this idea in mind, we are able to construct the model from the patient. Which is the particularity of the prostate? So tumor cells normally secrete PCA. So when we have cancer, this PCA that is created normally is produce a leakage of the PCA in the tissue, and this PCA go to the blood flux. So we need to simulate this process that is physically what occurs when we have a tumor. So for that, we construct a model that on the one hand, we have three main variables, the PCA at the tissue level that we simulate with a classical reaction diffusion equation, where we have the diffusion of the PCA, the production of PCA by tumors, the degradation of this PCA, and also this is the K-trans is the variable that we have obtained from the patient that is defined as a variable that regulates the flux between tissue and blood. So we can estimate how the PCA flux moves from tissue to blood. This equation is an ordinary differential equation that where we evaluate how the PCA in the blood, in the serum. So we evaluate this PCA in all the domain, but also keep in mind the decay of this. And finally, we have the tumor cell concentration, that how the tumor cells are proliferating with the time. With this idea in mind, of course, we have some parameters that we have to determine that are alpha P, gamma S, and alpha T. But we have additional data because we know the PCA concentration in the blood with the time. In the patient, we have the initial geometry of the tumor, but later with the time, we can follow the PCA concentration in the blood. So it is the information that we are going to obtain because normally the clinicians use this data for control the evolution, the progression of this tumor. We are going to simulate two different cases, patient 1 and patient B, A and B. And additionally, we are going to incorporate a fraction because all the tumor cells can see, but we are going to incorporate the fraction of proliferating tumor cells because from cellularity, we have all the concentration. We assume that a ratio of all these concentration are tumor cells. And this neural network is going to fit, again, we are going to use a neural network that is going to fit this parameter. But additionally, we are going to use more parameters that I'm going to explain now how the neural network works. So for that, we consider the specific geometry of the prostate of the patient. So we have a digital twin of this prostate, where in this prostate we have the tumor. We then take into account the 3D spatial contribution and the geometry. So the process is the machine learning is learning how is the distribution of the PCI dynamics with the time. So we are going to construct the model that I have explained. This is the process of calibration. So we have a physics-based model based in these parameters that I have justified, the PCA production rate, tumor growth rate, and the decays. And we are going to reproduce the patient-specific tendency. So for that, again, we construct a neural network that is based on the physics model that is based in continuum equations. But additionally, we keep in mind, we fit our model with the data from the clinicians. So these data are again feeding our neural network. So we simulate different conditions to fit again, to have an error that we have to reduce. So at the end, this is the PCA, this is the initial distribution and how is the tendency at different time points. And this is the error that we would like to optimize. This is the error that we have. Of course, if we have also the volume of the tumor, we have more data to validate our model. So this is the first case that we simulate. In this case, we have the tumor in the initial at the diagnosis. This is where we construct our model. Of course, then at diagnosis, we have the real tumor volume. And later, at the first follow-up, after 393 days, we have the prediction of our model and the SAT value. of course, we can see that our prediction, the volume, the fitness function is fitting the volume and the PCA. So, of course, we also can evaluate that this is the Bosel frequency that we have. And this is the prediction and the validation of the model. So, in this case, our model has been validated both with the volume of the tumor and with the PCA taste. This is in the patient I. In the patient B, we only have the volume and the diagnosis and the volume. This is where we construct the model, and we can observe again that our model is able to is validated, where we have a good agreement between the volume at day 710 and the PCA. So the predictions are quite good. Is it the validation? Sorry, not the next. We are going to see what are the future prediction of the model. So for that, we can observe that the different, this is the values that we have obtained for the calibration and validation. Of course, we can observe that the PCA production is much, production rate is much higher in patient B than in patient A. But what is more relevant, the tumor growth rate is much relevant in patient A in comparison with patient B. So that means that patient A is a high-risk tumor in comparison with patient B. We obtain the follow-up one is where we have the data, but now we are going to predict what happened the next days. What happened in follow-up 2 and 3? What happened later? So it is, we have some data from the patient, but other are numerical prediction. For example, in the case of patient A, we can observe that we have the tumor volume. This is where we validate, but these are predictions. And our predictions are quite good. So we don't use this data for fitting, for training the network. This is a prediction from the neural network. In the case of the PCA, in the case of the patient B, we can observe we don't have the volume but we have the data from the PCA, and we can observe that our prediction from our neural network is also quite good. And this would be the volume of the tumor prediction. Here we can see the numerical result of this simulation. This is the initial value of patient A, and we can observe this is the prostate, and here we can observe the tumor, and this is the initial value of the tumor, and we are going to see how is the process of growth of this tumor, where we have seen that our model predicts the evolution of the tumor and also predict the evolution of the PCA. This is a case patient A, and it would be the simulation corresponding to patient B that you observed. This would be the evolution of this graph. Of course, there are different tendencies, and each patient have different parameters because we create a specific model for each patient. So I think that I'm going to finish with some conclusion. Perhaps I have done a quicker presentation, sorry, but in this way we can have more time for questions. So I would like to say that in this case, we have focused in modeling tumor growth. So tumor growth presents several challenges. I have to focus in some of them, but in my opinion, always that we focus, we have to try to define the modeling strategy according to the data that we had for validation. This is what we have tried to do. So data is what is driving how the model has to be constructed. So the data is the most important for how is going to be the validation, and this is going to define which is the best strategy to model a specific problem. So I have shown two examples. On the one hand, we can observe tumor organoid. That is an excellent platform. We can observe that even in the same condition, each organoid presents a different behavior. So each organoid is different, but we can simulate with our approach any kind of cystic organoid. In the case, we also use examples. In the first case, we use examples where an agent-based approach, a discrete approach is used. However, in the second case, in the pattern-specific models, where we have data from patterns, we use a continuous approach. We use a quantitative description of nutrients, vascularization, and how is evolved the tumor. Predictive models are very relevant for evaluating the progression of the tumor. So it's important validation, but also we need to have a predictive capacity to evaluate the progression of the tumor. And finally, in my opinion, my main conclusion is that we need to work in the integration and combination of experimental data with numerical models. When we do that, we clearly are able to create more robust models that also will have more predictive capacity. So to finish, I would like to thank all the members of my group. Also, I would like to thank the funding institution from we received this presentation is mainly focused in two projects. On the one hand, ProCarinate project, that is a project funded by the Spanish Ministry of Science, and later there is is the part mainly focused in the clinical patients, and also the part of tumor organoid that has been funded mainly by ICOMIS, that is an ERC project, an advanced grant. And of course, all this project has been done in collaboration with other groups, with the company Kibin, Lafay, ANSYS, Sheffield, Bologna, and CIMA in Pamplona. Thank you very much. And so with that, I finish my presentation.
Thank you, Professor. That was a very insightful talk. We can now open the floor for any questions. If you do have a question, feel free to raise your hand, and Goran can unmute you, or feel free to post in the question chat. I guess to kick things off, I do have a comment and a question. You know, I think you've definitely persuaded us that you've done a very robust validation, and I would hope even clinicians would be persuaded by the validation as well, that, you know, the models are working, you know, as intended. I could definitely see the clinical utility of something like this being adopted in, you know, a clinical setting. I was curious what your thoughts are from the regulatory perspective, EMA or FDA. You know, what are the barriers for adoption for a, you know, neural network computational model such as this for prostate cancer?
Sorry?
Yeah, I was curious what your thoughts are from the regulatory perspective, like the FDA or the EMA over in Europe. What are the barriers for adoption for a computational model like this for use in the clinical setting?
Yes, okay. It's true that here normally the doctors, the clinical, have this data, okay? But not all the cases are good for simulation. So normally we need to select the cases because not always all the information is completely available. So for our model, it's very important that we are good quality of the images to obtain, above all, the cellularity distribution, also the vascular. So for doing the estimation of the vascularization and obtain the K-trans, that is the variable that we are estimating from the image, we need to have some reconstruction. Okay, if you don't mind, I'm going to present. The most difficult is normally, in the case of the clinical data, here. Normally, we need this, we need to have this curve. That is, normally we have a contrast agent, and we need to obtain this curve that normally this contrast agent is used in MRI, and to have this, to estimate the vascularization, we need many points in this curve. If we don't have this curve very well defined, it's very difficult to obtain the vascularization, because the vascularization of this, to estimate the vascularization of this problem is an inverse problem that normally is very bad condition, okay? Very ill condition, okay? Is it your question, more or less?
Yeah, more or less, yeah. Appreciate it, thank you. So we have two questions in the chat. One is from Luigi. They say, thanks for the nice presentation. I may have missed it, but is intercell variability accounted for in your model? Is it something you can characterize experimentally?
Okay, in our model, my question is in the continuum or in the discrete model? In which model is asking, you know?
It's just saying in general. Luigi is just saying in your model. Luigi, can you clarify? If you have a mic, we can also unmute you as well.
Of course. Okay, it depends on the case, okay? Of course, variability is always, we are going to have a variability. In the case of the first model, in the case of the tumor organoid, variability is in the same in the model. So our model also predicts variability. So the variability that we can measure in the tumor organoid, for example, in this case, we select 4 organoids, is our model is able to predict this variability, okay? So you can observe here, we have 4 organoids, but we can quantify the variability, for example, in the number of cells and the lamin volume. And our model is also able to predict this variability. So depending on how the process. So we here try to understand which is the source of this variability. Okay, so the source of variability in this experiment in particular is due because some cells proliferate or secrete fluid at different time scales. So because each individual cells have a different clock, so depending on this, the differences on variability are due to this. Of course, if later we move, for example, to the case of the continuum model, of course, in the pattern there is variability, and the measurements of PCA is very difficult. But in this case, we don't incorporate variability itself because our objective was to predict the tendency of the evolution of the PCA. But of course, PCA is quite variable. So depending how you do the measurements, so in this case, in the model, in the case of the clinical case, our objective was not to predict the variability, was to predict the tendency. So at the end, the variability of the model have to be defined depending on the prediction or the validation that you are aiming to do. So if you are going to your objective is to predict this variability, we have to incorporate variability in the model. But if we are not, we don't have this objective, we don't have to predict that. Therefore, it's not necessary to incorporate variability. I don't know if I reply, but I have tried.
So Luigi has a follow-up question. They say, okay, so the model is deterministic rather than probabilistic. Thanks. Oh, okay, it was just a comment. Thank you, Luigi. Next question is from, go ahead. Go ahead, Dr. Garcia Aznar.
No, okay, what I said is, in the first model, in the discrete model, our model is probabilistic. So because the cells have different options to select the process. However, of course, in the second model, in the continuum model, is deterministic. But in the first model, it's more probabilistic. But of course, in the case of the continuum model, we can incorporate a probabilistic model easily. The question is, it wasn't our interest, because our interest was to predict the tendency of the PCA evolution. Okay.
So next questions from Georgios. They say, many thanks, Jose, for the excellent presentation. Have you thought about simulating the response of prostate tumors to various treatment modalities?
Yes, of course. Our idea here is not, we have not simulated treatment, but our idea is to simulate treatment. We have simulated treatment in, for example, in neuroblastoma with chemotherapy, and we can predict also good results. Of course, we need validation, and we need also to estimate how it is prediction, but the treatment is possible to be done. Above all, if the model is based, for example, in chemotherapy, because chemotherapy is distributed through the vascularization of the tumor. And we have a model that is quite representative of the vascular system of the tumor. Therefore, all the treatments that are based in chemotherapy, we are able to predict the evolution of the tumor. If he's interested, he or she can send me an email, and I can send him a publication in this direction.
Great. And then somebody in the chat asked, are you able to share the presentation, and also will the recording be available? I can answer the last question. Yes, the recording will be available. Goran, correct me if I'm wrong, likely within the next week or so on YouTube. So we can send it.
Yes, I'm going to share the presentation to the VPH. I send a PDF, but also I am available to have a meeting, or if you have some doubt, or to solve this doubt individually. I have no problem for that. Send me an email. Of course, I send you to the organizer. I will send you my presentation, but of course, if you would like to do an individual meeting, and I'm able to do it.
Great. So Moin asks, they say, thank you for the clear presentation. I want to know that if your predictive model is based on physics-informed neural networks, specifically, do you calculate the loss function and train the model by directly incorporating physical laws into the learning process?
Okay, I haven't understood the question. So if he means that, of course, at the end, the neural network is fitting the result that we have. Of course, we need this data. If this data is not possible to fit in. So, sorry, can you repeat the question? I don't understand.
Sure. So essentially they're asking, is the predictive model based on physics-informed neural networks? And do you calculate the loss function and train the model according to these physical laws?
Yes, of course. The idea is with the physics laws, we make predictions with the different parameters, and we are training the network to find which is the combination. Okay, depending on the case, because the neural network works in different way. Okay, in the first case, more than defining the parameters, we are defining if the cells probabilistically are doing a process or another. There are cells that proliferate quicker, other cells secrete more fluid. So we are estimating, the neural network help us to define this. However, so in the first case, if we give the lumen volume and the number of cells, that is the data, the neural network is able to predict which kind of organoid is going to be formed. Okay, so it's one strategy that it works in this neural network works in this way. However, in the second case, the neural network works in a different way. Our idea is we have some physics law that are deterministic, okay? And in this deterministic, we are fitting the parameters to obtain the validation and to validate the best results, fitting both the volume and the PCA. So it's clearly two different examples.
Great. Do we have any other questions from the audience?
I do not see raised hands nor questions in the chat.
Okay. Well, I think it's, are we okay to close up, Goran? You can do a closing remarks. And great. Yep. So on behalf of the VPHi Young Sciences Committee, thank you, Dr. Garcia Aznar, sharing your expertise and also very engaging discussion on your topic. As a reminder, the recording and any permissible slides will appear on the VPH website as well as YouTube. Look out for our next keynote presentation soon. Check your inbox as well as VPHi LinkedIn. And until then, stay curious and have a productive day.
Okay, thank you very much for your invitation.
Thank you, everyone.
Thank you.
Have a nice day.